Hello, I'm David Tricoli from the University of California at Davis, and today I'd like to talk to you a little bit about our protoplast-mediated gene editing system for disease resistance. In this work, we are using the CRISPR-Cas9 genome editing system, and this is a method of very specifically editing DNA. It's being used in all areas of biology now, including human healthcare, and we are applying this CRISPR-Cas9 gene editing technique to try to improve grapevines. The system uses a protein called Cas9 which can cut DNA, but we don't want it to cut DNA in any random region. We want it to cut DNA in a very specific region. So we link the enzyme to a guide RNA which will escort the enzyme to that specific gene in the plant that we want it to cut. We are delivering the Cas9 system to grape cells and then regenerating whole plants from those grape cells. The difficulty, however, with plant cells is they have a cell wall, and we cannot get the CRISPR-Cas9 complex through that cell wall. Therefore, we have to take the cells and digest the cell wall but we're using enzymes to generate what are called plant protoplasts. And the plant protoplasts are basically a plant cell without the cell wall. Now we can easily deliver the CRISPR-Cas9 into the cell now that the cell wall is gone. Once we have the protoplast and we have delivered the Cas9, the critical part is being able to form a whole plant back from those plant protoplasts. And our lab has spent a number of years developing the system for grape, where we can take individual plant protoplasts and within about 6 months get whole plants back from those protoplasts. So here's a cartoon of those protoplasts, and we can just mix those protoplasts with the Cas9 enzyme and the guide RNAs, and the guide RNAs will traffic the Cas9 to the nucleus of those cells into the very gene that we want edited, and will make the cut of the DNA in that gene. To demonstrate the system, we first targeted the gene in the plant that's necessary for making the plants green, for producing chlorophyll. And we could deliver the Cas9 protein to the protoplast, and if we edited that gene, we would turn off those genes that are responsible for making the plant green, and plants would develop as white albino plants. And here you see an example of that, of two plants there where we have knocked out the plant gene responsible for making chlorophyll, making the plants green, and these two plants are white and are albinos. Now that we had demonstrated that we could edit plant genes by creating albino plants, we turned our attention to susceptibility genes. Now, susceptibility genes are genes that are plant genes that, when expressed, they help facilitate the infection of the plant by a pathogen. We are using our protoplast-based gene editing system to turn off 3 susceptibility genes that are called MLO6, MLO7, and MLO11, and we believe that these genes are responsible for allowing powdery mildew to infect grapevines. We have produced a number of plants with various edits in these genes, so we have knocked these genes out so the plant no longer expresses those genes, and we hope that by turning these genes off, we will get enhanced resistance to powdery mildew. The plants seem perfectly normal at this stage, and we are cloning them to make copies of them, and then we will inoculate them with powdery mildew spores and see if we have increased resistance to the disease. It's important to note that since these plants are made by gene editing of the genome and not by insertion of foreign DNA, they are not considered GMOs by the USDA-APHIS regulatory system, and therefore are not considered transgenic within the United States. We hope to have screening results within the next 3 to 4 months, and I hope to come back and give you an update of our progress. Thank you.